Hello, everybody, and thank you for joining us on this nice October morning for our Precisionary webinar. So I want to introduce Katherine Pizano, who is a graduate student from the lab of Dr. Joshua Singer at the University of Maryland College Park. And Katherine's research journey has taken her from Princeton University, where she explored molecular bio and in vivo calcium imaging techniques, to a post-bac fellowship at the NIH, focusing on neuropeptide input to gonadotropin releasing hormone neurons. And in her current role, Katherine investigates how PACAP, neuropeptidergic, oh, I can't pronounce this word, modulation by ipRGCs impact synaptic communication in areas like the SCN and PHb. She uses whole cell patch clamp recordings in ePhys in behavioral paradigms. So today, Katherine is going to guide us through slice preparations for ePhys recordings, showcasing optogenetic mechanisms and protocols that she's used. So please join me in welcoming Katherine to our webinar stage. So with that, over to you.
Yeah, so hi everyone. Thank you so much for that introduction. As was mentioned, so today I wanted to give an introduction to how our lab uses slice physiology to better understand the synaptic transmission occurring both in the retinal circuitry as well as more recent work in the brain. So to start, why do we study the retina? Well, the retina is a model circuit that has been extremely well characterized and has very known physiological functions. It is capable of transforming visual cues into electrical outputs to the brain that allow us to understand what we see in the world around us. So we tend to think of the retina as being composed of two main pathways. There is a vertical excitatory pathway and a lateral inhibitory pathway that modulates the signal from that vertical pathway. So when light first enters the eye, it's absorbed by these photoreceptors, namely rods and cones. And while rods and cones assess different image properties, they are thought to work in similar ways. These photoreceptors release glutamate tonically in the dark. It's also worth mentioning that there are several subtypes of cones, all with distinct spectral sensitivities, although it's known that rods tend to be more light sensitive. Now, synaptic output from these photoreceptors is eventually passed on to retinal ganglion cells by vertical excitatory pathways. Rods and cones make these synaptic contacts onto roughly 12 different types of bipolar cells in this inner nuclear layer, which feed into two parallel pathways for image processing, the on pathway and then the off pathway. These bipolar cells can be characterized in that they're glutamatergic interneurons, which provide excitatory input onto retinal ganglion cells. And bipolar cells in the on pathway are known to detect increments in luminance, while bipolar cells in that off pathway are signal decrements in luminance. The polarity of these bipolar cell responses is determined by the glutamatergic or glutamate receptors it expresses. So the unique combination of these receptors expressed by individual types can contribute to distinct downstream signaling in these pathways. Now, apart from this vertical excitation, there's also lateral inhibition in the retina, involving input from both horizontal cells, shown here, as well as Amerkin cells. Horizontal cells are thought to help tune signaling that is done from these photoreceptors to bipolar cells, while Amerkin cells help tune signaling from these bipolar cells to ganglion cells. Now, evolutionarily, we know that rods are newer than cones. In many animals, apart from mammals, rods and cones actually signal to the same bipolar cell types. However, in mammals, including humans, the majority of rods synapse to a specific type of bipolar cell that doesn't contact ganglion cells directly. This additional type of cell, which I didn't mention before, is called the rod bipolar cell, and it receives input from rods alone. Shown here in the figure on the left, it's part of a distinct pathway that is dedicated to processing this rod output. And the synaptic physiology of the circuit has been of interest to our lab due to its unique characteristics. So what makes this pathway so unique? Well, it's been reported. So in humans can respond and detect very low light thresholds of only a few photons. And this implies two things. So first, rods must have the capability or must be responsible for generating responses to the absorption of single photons. And second, single ganglion cells must also be able to respond to these single photons being emitted. And it's believed that all of this is occurring in this rod bipolar pathway. So taken together, these two observations suggest that within this pathway, there must be mechanisms by which to amplify very small signals, as well as get rid of synaptic noise. So there has to be a mechanism for strong gain control that facilitates this transmission that I've been describing of single photon responses from rods. And our lab has been interested in studying the synaptic mechanisms by which this is possible. So work from former lab members, including some of the retinal physiology I will show later on, has focused in understanding the unique attributes of the circuit, including characterizing the intrinsic properties of these rod bipolar cells, tracing their synaptic outputs, as well as looking at how information is conveyed throughout the circuit to allow for synaptic gain control. So while the lab has traditionally studied these synaptic transmission and circuits in the retina, we've also more recently become interested in looking at the communication that occurs from these ganglion cells to brain areas involved in non-image forming vision, exploring topics like light-dependent modulation of behavior. My own interest specifically during my PhD involved looking at retinal output to both the suprachiasmatic nucleus, or SCN, which is shown here, and the perihabenular nucleus, or PHb, which is up here. Two pathways, which I will also provide some preliminary data later on. So up to now, I've explained how information from photons is conveyed throughout the retinal circuitry in a canonical visual pathway. And regardless of the circuit in question, all information, as I've mentioned, is integrated at the level of these retinal ganglion cells. From there, these ganglion cells make synapses to brain regions involved in image-forming vision. But now there are over 40 known types of ganglion cells that can help process this visual scene. And of these, more recent work has found that there is a special type of ganglion cell. It's called the intrinsically photosensitive retinal ganglion cell, or ipRGC for short, which is essential for non-image forming vision. So these ipRGCs predominantly receive input from the on pathway, although this can vary among subtypes. And ipRGCs are unique because, unlike other types of ganglion cells, they have phototransduction capabilities thanks to the expression of melanopsin. Similar to rods and cones, ipRGCs have the capability to convert photic input into electrical signals. So these ipRGCs encompass a morphologically and functionally diverse type of at least 6 subtypes, some of which I show you down here. And the variability of ipRGC responses can be really appreciated and influenced by two things mainly. So one, the intrinsic properties of the melanopsin itself, and two, by extrinsic factors like dendritic stratification. Now, in addition, ipRGCs project to several anatomically distinct brain regions that regulate different types of behavior. And today we will discuss how ipRGC projections to the SCN and PHb can affect some of the things I've mentioned. So these two areas help regulate circadian rhythms and mood respectively, and they're unique because they receive retinal input almost exclusively from these ipRGCs. Also, a key attribute of these ipRGCs is their sustained firing rate during long periods of light exposures. Experiments have shown that even under a 10-hour light stimulus with varying intensities, ipRGCs can remarkably continue to fire throughout the entire time. But even among the better studied retinal hypothalamic tract, that is the ipRGC to SCN pathway, shown here, there are very few electrophysiological studies looking at how information at the level of ipRGCs in the retina is conveyed to these retinorecipient neurons. as has been previously mentioned in other talks. So we know that ipRGCs release a number of neurotransmitters, including glutamate, GABA, and the neuropeptide PACAP, or pituitary adenylate cyclase activating peptide. An example of excitatory input being conveyed from ipRGC to SCN neurons can be seen on the figure here on the left. So with the aid of optogenetics, researchers here were able to stimulate ipRGC fibers in the SCN and record from the postsynaptic neuron. So here is an evoked EPSC showing evidence for glutamatergic input. It was also discovered that ipRGCs can convey that inhibitory GABAergic input to some portion of these SCN neurons as well, as shown by this figure here. However, even less is known about peptidergic transmission onto these retinorecipient circuits, especially how PACAP can shape the pre- and postsynaptic responses within these pathways. And this is surprising because PACAP is found in large quantities within the SCN, both in presynaptic ipRGC fibers, as this figure above shows, as well as diffused throughout the region, as shown on the bottom left figure. What's more, PACAP could also be integral in shaping responses in other ipRGC projection regions, like the PHb, where there seems to also be some sparse labeling of the peptide. So it's possible that PACAP could be playing a role in fine-tuning individual synaptic input to better convey information on light intensity, duration, that can then allow for quantifiable changes to circuit-wide behavioral responses. PACAP release most likely depends on a combination of both the intrinsic properties of ipRGCs along with extrinsic cues, and studying this PACAP modulation, like our lab has started doing within these synapses, can help us gain a better understanding on how these neural connections can lead to these established behavioral effects. Now, of course, there is literature that studied the neuropeptide to some degree, and most of the literature involving PACAP does focus on this retinal hypothalamic tract. And papers can mainly be divided into two categories. So first, those that add exogenous PACAP in slice preparations, and then several papers that look at germline knockouts of the peptide to look at circadian behavior. So PACAP is known to be part of this VAP glucagon superfamily. It's also known to bind to the PAC1R receptor, and it activates adenylate cyclase. Now, if you look at examples of papers that add exogenous PACAP, you can see that when you add exogenous PACAP and record from SCN neurons, you can have increases in spontaneous EPSC frequency, shown in this figure here. So addition of PACAP, there are more spontaneous EPSCs, as well as increases in amplitude. We can also see phase shift advances. So an example of that is in this diagram below. So if you add PACAP at circadian time 6, the peak firing rate actually gets shifted forward, so it occurs earlier than it would otherwise in the control condition. If we move on then to look at some examples of papers where you use PACAP knockouts, these studies tend to show phase shift deficits or impairments in photoentrainment. So in this example above with phase shift deficits, what the experimenters did here was that they exposed both wild type animals and these knockout animals to light at different light intensities. So at 10 lux, 20 lux, and 100 lux, as well as different time durations. So either for 5 minutes or for 30 minutes. And what they found was that at brighter light intensities, knockout animals have failed to have a phase shift compared to wild types. Moving on, if you then look at impairments and other impairments in photoentrainment, here is an example where the light cycle was manipulated to include a 6-hour advance or delay of the light onset. And it was discovered that these PACAP null animals take significantly longer to reestablish entrainment, but only for this phase advance condition. So what that means, in other words, it took these PACAP knockouts, this black bar, roughly 4 more days for about 50% of the animals to re-entrain to this new light onset. It's also worth mentioning, though, that while these PACAP knockout studies have discrepancies in photo entrainment, if these animals are left under constant darkness, they show no difference to their wild-type counterparts when it comes to things like locomotion and periodicity under constant darkness, which suggests that their internal clock is functioning properly, or at least is being compensated for. So the problem really seems to be with the integration of external light cues. And now, while all these papers provide us with a lot of valuable insights onto the role of PACAP, studies tend to lack one of three things. So one, looking at the SCN, there tends to not be any cell type specificity in these studies. Two, germline knockouts might interfere with retinal development. And three, many of these studies fail to look at how ipRGC presynaptic input can affect these responses. So for today's talk, I will want to focus on a few key examples, both in retinophysiology as well as retinorecipient circuits, to look at first answering the question, how do the properties of transmission in this rod bipolar pathway influence visual signal coding? And then how can PACAP shape synaptic responses in the SCN? If time permits, I also want to show some preliminary data, behavioral data on PACAP's potential involvement in the PHb. So to answer these questions, our lab employs electrophysiology. For our retinal patch recordings, we tend to use whole mount retina, so no slicing is required. But for our brain recordings, we do use the compressome. We have a slightly older model, as shown here, but I found that the compressome is a quick and easy method to obtain slices compared to other vibrotomes, which usually take longer for slice collection. And this is an important factor to consider when looking at the health and quality of the slice, as well as overall prep time, depending on what protocol you use. And here on the right, I just wanted to include a photo of one of the rigs in our lab. So what exactly is our protocol for a patch clamp recording? A small overview for the retina, there are less steps. So we tend to make an Ames media that we use for patching, which is used to support the overall retinal tissue health. We then collect the eyes over our animal of interest and dissect out the retina. To do so, you have to be careful of not damaging it. So typically a small hole can be inserted around the sclera, from which you can open up the eye and extract the lens, being very careful, again, like I mentioned, not to damage the retina as you take it out of the eye cup. You can then leave your retina whole mount in Ames until you're ready to patch. Now for our brain protocol, there are slightly more steps. Our lab, like many other labs, have adopted this NMDG slicing protocol developed by Jonathan Ting, which is meant to even further extend the health of the slice. One way this protocol differs from other slicing solutions is that instead of, for example, having the holding and recording ACSF be the same, they distinguish between this initial recovery incubation shown here after slicing that they've found to be very important. Also, this NMDG solution allows you, instead of traditionally, you have to patch fairly young animals, 3 months or younger. And with this protocol, they've shown that you can patch onto cells in animals that are 6 months old, if not even older. And this recovery step that is important within this protocol is known to be low in sodium. It's low in calcium and high in magnesium, from which you then slowly add sodium in what they call the sodium spike-in over incremental time points, depending on the age of the animal. So you can adapt it to your needs. And we've incorporated this protocol pretty faithfully in our lab. So for all our brain recordings, we start by prepping our solutions. So that NMDG HEPIs is our cutting solution, as well as our protective recovery solution later on. And then we have our HEPIs holding as well as recording ACSF. Then after our brain dissection, we can use the compressome to slice our brain slices. We typically use 250 thick slices. And to slice our slices, so we gently mount our extracted brain onto the specimen holder, secure with adhesive glue, and embedded in 2% agarose. And after recovering our slices, we move them to the recovery chamber at 34 degrees, where we can begin our sodium spike-in. And the final step before we can begin patching is to transfer all these slices into our HEPIs holding ACSF for an additional hour. And here I just wanted to show some examples of what we're seeing under the rig. So for retinal whole mounts, here is an example of a fluorescent ipRGC cell body after a retinal injection was done. And then on the bottom is an example of one of our SCN slices. And this is the recording pipette. And to the right of that, we can see ipRGC fibers from that same animal surrounding the region. From there, we can do, of course, current clamp recordings looking at action potentials, or voltage clamp recordings to look at evoked excitatory postsynaptic currents or inhibitory currents from these our retinal recipient neurons, which I will expand on momentarily. So now, with that background, I want to bring into focus how our lab uses these techniques along with optogenetic approaches to answer specific questions on synaptic transmission. First, by looking at the work by previous lab members within this rod bipolar pathway, and then more recent work looking at peptidergic input in retinal recipient circuits. So within the visual scene, light intensity varies a lot. There are areas, if you look at this image here, there are areas that are uniformly dark, as depicted within this blue square. There are some areas which are uniformly bright, as the sky in this photo, shown in this green square. And then there are areas that have a lot of spatial contrast down here in this red square. And what essentially this means is that these areas have bright and dim stimuli that are close together in proximity. This is further expanded on in the bottom schematic, showing how the gain of signaling is adjusted to light intensity. So here, these graphs show an example of the light intensity versus response relationship. And one thing to note, for example, is how the slope is steeper for dim stimuli over bright stimuli, which can be interpreted as meaning that areas of lower contrast have responses with higher gain, while these areas with a high contrast have responses with lower gain, so that the response of the encoding cell, the ganglion cell, doesn't saturate. However, a question there is in the field is that we know that these retinal ganglion cells can only change their firing frequency over a few orders of magnitude. So we have to ask, how can the retina encode for stimuli that varies more than the firing rate of these ganglion cells can change? And one way the lab has studied this is to use an approach that borrows from linear systems analysis. So this approach will allow us to describe the transformation that's occurring between the signal and response in 2 steps using a linear nonlinear model. Now, without getting into too much detail on the model itself, we can use it to both describe and quantify these changes in the gain of signaling within neural circuits. And in the following slides, I'm going to highlight how we can use optogenetics to study these retinal circuits and begin answering these questions. To do this, we're going to employ a white noise stimulus, where we can then measure the transformation that's occurring from stimulus to response of the selected cells in the pathway. But before we do our white noise stimulus, we first wanted to confirm that we can drive responses in the rod bipolar pathway using channel rhodopsin that are comparable to photoreceptor-driven responses. While cone bipolar cells signal directly onto ganglion cells, rod bipolar cells instead signal to ganglion cells indirectly via this A2 amacrine cell. And it's this A2 amacrine cell that then makes excitatory or inhibitory outputs with bipolar cells. And then these bipolar cells, from there, these on and off cone bipolar cells convey their signal to ganglion cells. Because we know that this on alpha ganglion cell responds to visual thresholds that are very sensitive, they get a lot of synaptic input from the rod bipolar pathway, which makes this a good cell to patch onto to see what's occurring. So first, what we did was use a dim light stimulus, shown here, to stimulate rod input and record from both these A2 amacrine cells on the right, as well as the downstream response of an on-alpha ganglion cell on the left. And when we do this, you can see that both the A2 amacrine cell and the on ganglion cell respond to our stimulation. Now, traditionally, our lab has used retinal slice preparation in a lot of our studies, and as optogenetic manipulation slowly became more common in the field, we wanted to repeat these experiments to look at how signal transmission by stimulating cell types directly. So we could then use this PCP2 Cree AI32 animal to stimulate these rod bipolar cells directly with this channelrhodopsin while blocking photoreceptors. And as you can see, similar to stimulating the rod bipolar cells with real light, when you use channelrhodopsin, you can drive comparable responses in both the A2s and on ganglion cells. So this proves that we can stimulate these rod bipolar cells. And from here, we can use this method to then ask further questions on how the signal is being transformed in the pathway. So with this as our baseline, our lab did 2 sets of parallel experiments looking at synaptic input onto the on-alpha ganglion cell down here in the circuit. So going back to this white noise stimulus, we can compare ganglion cell responses when stimulating either, like I mentioned, the rod bipolar cell, or further downstream by stimulating the type 6 cone bipolar cell. And this will allow us to know whether the ganglion cell response is more dependent on the rod bipolar or cone bipolar output. When we stimulate that rod bipolar cell with a white noise stimulation, depicted on the right, we notice that there is evidence of contrast adaptation from our recorded on ganglion cell. And supposedly, so the response that we are seeing down here reflects the rod bipolar cell giving input to this A2 amacrine cell, which in turn synapses onto the on cone bipolar and finally makes its way to the ganglion cell we record from. And you can observe that the response depends on the contrast of the stimulus. If we then do the second experiment, which is stimulating that on cone bipolar cell and recording from those ganglion cells, we see that this effect disappears. So here, unlike before, the gain of the response is not affected by the variability of the stimulus, and there is no contrast adaptation. So with that, moving on to the second part of the talk, I now want to focus on recent work that I've done looking at retinorecipient circuits. So previous work in the lab showed that we can also use this optogenetic stimulation to stimulate ipRGCs and study retinal projections to non-image forming visual areas. So figure C here shows an example of this stimulation being given to axons and being given to these ipRGC axons. And then you can record excitatory postsynaptic currents in cells within both the OPN or olivary protectile nucleus and the PHb, 2 regions that are known to be retinorecipient. And in being able to compare these responses from both retinorecipient and non-retinorecipient cells, our lab, as others have, can begin to decipher how this ipRGC input influences the local circuit transmission within these first few synapses in the brain before being sent to downstream pathways. So first, in looking at the ipRGC to SCN pathway, to better understand how PACAP might have population-specific effects here, we can use the CRFR1 GFP mouse line that's been particularly useful to us. And we want to use this mouse line because simultaneously targeting SCN populations while stimulating ipRGCs is challenging. And this line allows us to circumnavigate a common issue when trying to study synaptic transmission among specific cell types. And that is that when looking at ipRGC synaptic input, you have to forego one of 2 things. So you tend to have to forego presynaptic manipulations. So you can use a mouse line or mouse model that targets a specific SCN population, like I've mentioned before, but then you tend to be unable to stimulate any presynaptic ipRGC input. Or you can instead choose to stimulate ipRGCs using an OPN4 line, but the trade-off being unable to distinguish the cell type you're recording from until after the experiment. And we know that the SCN itself is very heterogeneous, so it contains at least 5 known cell populations, not to mention that there's also subpopulations that may have functional relevance themselves. So luckily, this CRF GFP mouse line can help alleviate some of these problems. So what is CRF? So we know that CRF helps regulate stress responses, and deficiencies in the CRF lead to decreases in anxiety-like behavior as well as impaired stress responses. We also know that SCN VIP neurons synapse onto these CRF neurons in the PVN to help shape some of these responses. Moreover, there have been previous transcriptomic studies that confirm, so we're interested in PACAP, and these studies have confirmed that the PAC1R receptor is only expressed postsynaptically in the VIP GRP population, one of the many subtypes of the SCN, which also happens to be the only population expressing this CRF receptor 1. So here we did some immuno to confirm that further. So in the first few figures is a VIP immunostaining of a coronal section of the SCN in red, and then in green is CRFR1 positive GFP cells. And the merged image shows an overlay of both, with the arrows depicting a single cell, suggesting that a portion of these CRF cells do indeed co-express VIP. The bottom image now shows a similar staining, except now we are seeing for GRP or gastrin-releasing peptide, one of the other main cell types in the SCN. So we hypothesize that a portion of these CRF neurons that we will record from overlap with these GRP VIP neurons. And these neurons contain the PAC1R receptor postsynaptically, as we show here in this modified Venn diagram. So how are we going to experimentally test this? So to ask if PACAP is involved in this pathway, we can cross an OPN4 animal to a CRFR1 GFP mouse, to the CRFR1 GFP mouse. And then after obtaining our heterozygotes, we can then inject a Cre-dependent crimson virus into the retina to selectively stimulate ipRGCs with a red light-driven channel rhodopsin. Then we wait a few weeks and we can go ahead and perform our patch clamp recordings, where we can now record from CRFR neurons under voltage clamp conditions while being able to optogenetically stimulate our ipRGC fibers. So the first thing we wanted to do with this paradigm or this experiment was to characterize just the overall responses of these cells to this optogenetic stimulation. And we confirmed that these CRFR1 cells are in fact retinorecipient. Moreover, if we use a paired pulse protocol where we stimulate that ipRGC presynaptic fiber with 2 5-millisecond pulses, we can see that these cells display both synaptic facilitation as well as synaptic depression, although a majority of the cells we did record from showed this synaptic depression response. We also can note that after stimulation in the presence of 4AP, these CRFR1 cells exhibit asynchronous release, as depicted by the arrows. So now, to investigate the effects of PACAP on the synaptic transmission, we once again we can stimulate that ipRGC fibers, those ipRGC fibers in the SCN, and look at the evoked responses in postsynaptic GFP positive cells, both before and after the addition of a PACAP agonist, PACAP38. So on the left here are sample traces from our recordings after stimulation. And as you can see from the first graph, addition of this PACAP agonist leads to a decrease in the evoked amplitude response following that first pulse stimulation. On the right is a second depiction of the data. So here you can see our 5-minute baseline control followed by washing on this PACAP agonist for 10 minutes. And as in the first graph, you can see the effects PACAP has on that first pulse amplitude over time. So now this data suggests that PACAP depresses the ipRGC CRFR1 synapse, most likely through a postsynaptic mechanism, perhaps, but more studies are needed to confirm this. If it is postsynaptic, it would be consistent with previous studies or reports suggesting that these CRFR1 cells contain that PAC1R receptor postsynaptically. But next, we also wanted to investigate whether there could be a presynaptic PACAP-induced short-term plasticity in these cells. And to do that, we can use our paired pulse protocol. So on the left are more sample traces of this protocol, highlighting the variability of responses. So even though there has been evidence of short-term plasticity at the synapse, looking at the paired pulse ratio in this first graph, it seems that PACAP does not play a significant role here, although it must be said that given the heterogeneity of the CRFR1 cells, we suspect that PACAP could be leading to paired pulse facilitation in a subset of these cells, and this would become more evident if we increase our N. And the next thing we did was to look also at the mechanism, to look further at the mechanism of PACAP-induced responses. We looked at the amplitude of NMDA receptor EPSCs. Given that there's previous evidence to suggest that PACAP can enhance the magnitude of NMDA-evoked currents in some SCN neurons. So for this set of experiments, we held the cell at positive 40 in the presence of picotroxin to block any GABAergic input, and we analyzed the amplitude 50 milliseconds after the stimulation was given. Although it seems that in the cell type, there seems to be no significant change in NMDA current with the addition of PACAP. Now, because PACAP is known to bind to both the PAC1R receptor, which I mentioned before, which is specific for endogenous PACAP, but it's also known to bind to these type 2 VPAC1 and VPAC2 receptors that can bind both to PACAP and VIP with varying degrees of affinity. So we wanted to identify the receptor subtype mediating the observed PACAP responses. If the effects of PACAP on the first pulse, on the evoked amplitude are modulated by the PAC1R receptor, we would expect that PACAP-induced depression of glutamatergic responses to be blocked if we then use a PAC1R receptor antagonist. And indeed, that's what we observe as shown in this slide. So once again, on the left is a trace example from our paired pulse recording during both the PAC1R antagonist only condition, as well as when you add PACAP, when you wash on this PACAP. And then on the right, we quantify these results. So in the presence of the antagonist, there seems to be no change or no significant change in the normalized amplitude of our evoked responses, even when we add PACAP. And then the graph on the left shows the same results, except now we plot it over time. So these results suggest that PACAP works through the PAC1R receptor to mediate the decrease in amplitude observed from our stimulation. And then very last thing I wanted to mention in the last few minutes, I would like to go over one final set of experiments that we are currently working on in the lab with a focus on the ipRGC to PHb pathway. Now, apart from SCN-dependent pathways of mood regulation, there has been recent evidence that suggests that ipRGCs can also innervate the PHb to lead to light-mediated mood regulation. And these PHb neurons have been found to project to several mood centers in the brain and are very well positioned to decode these environmental light cues into behavioral outputs. However, the mechanisms by which ipRGC innervation can lead to the activation and regulation of these neurons is still in its early stages, but previous work from the lab and from our collaborators has shown that constant activation of these PHb neurons can lead to depressive-like symptoms under normal light conditions. So that's this T24 cycle of 12 hours lights on versus 12 hours lights off. While if you silence these PHb neurons under an irregular light cycle, you can block depressive-like behaviors. And briefly, this irregular light cycle, or T7, is used to model aberrant light, and it has alternating lights on and off of 3.5 hours. And it's been previously shown to cause depression or depression-like symptoms in mice. Now, if you look more closely at the intrinsic properties of these PHB neurons, it seems that these neurons have increased tonic activity under the aberrant T7 cycle. And it's also been shown that ipRGCs make excitatory synaptic contacts onto mostly GABAergic PHB cells, as opposed to glutamatergic cells that also exist in the PHB. So now, given that this photic input to the ipRGC PHB circuit has very well-defined consequences on mood, we thought it'd be a model circuit to further explore the functional relevance of PACAP in this parallel ipRGC pathway. So to better try to understand this question, what we want to learn is whether PACAP being released from ipRGCs can have any behavioral effects on the circuit, namely if PACAP signaling can affect these light-induced mood changes. So to test this, we first inject an AAV Cree virus into the retinas of PACAP flux animals, and this allows us to selectively knock out PACAP in retinorecipient pathways. And then after waiting a few weeks for viral expression, we can place both these PACAP knockout animals and their wild type littermates under either a normal T24 light cycle or that aberrant T7 light cycle that I briefly discussed before. After letting the mice be in these light cycles for roughly 2 weeks, we can then use mood assays like the sucrose preference test targeted at seeing if there is any mood changes between these animals. So with the sucrose preference test, mice, if they're not depressed, they tend to enjoy sweet things. So mice will prefer 2% sugar water over regular water, versus if there are any depressive-like symptoms in these animals, mice will have no preference to either the sweet sugar water or the regular water. And we suspect that when we knock out this retinal PACAP, it will actually be preventing disruptions to the ipRGC PHB circuit caused by irregular light cycles or light changes. And so here I'm showing you a sample image of what our viral injection looks like. And in the figure, you can see expression of TB tomato in the PHB. So presumably, wherever there's expression, their presynaptic PACAP has been knocked out in that region. And then if we move on to look at our preliminary data, you can see that so wild type animals, when placed under T24 versus T7, show a decrease in that sucrose preference. So percent sucrose consumption goes down, which is supported by the literature. Now, encouragingly enough, if you then look at our conditional knockout animals, these knockout animals under both light cycles show similar sucrose preferences. And this suggests that when you knock out PACAP, it seems to rescue that depressive-like symptom that is characteristic of mice placed under an irregular T7 cycle. So with that, hopefully I've been able to give a brief overview of how our lab uses these both optogenetic techniques along with electrophysiology and some behavior at the very end to better understand how local transmission, both in the retina and more preliminary work in the brain occurs. And with that, I just wanted to conclude the talk and then open it up for any questions. Thank you.
Thank you so much, Catherine. We had a question already during your talk that Dr. Singer actually answered, but because this is recorded, they can't see the chat. So I'm going to go ahead and ask it if you can answer it again, which is how is transmission in your experiments from photoreceptors blocked when you're doing your recordings?
Yeah, so it's blocked with the answer was with LAP4, which is an mGluR6 agonist, as well as ACET, which is a CAR antagonist.
Wonderful. I actually had a quick question. And by the way, everybody, I encourage you to unmute yourselves and show your video if you feel comfortable to ask questions. I had one where you talked about the retina mount and also brain slicing. Have you ever had experiments or need to section the eye as a mouse eye as a whole where you don't mount it, but you have to make sections? Yes, exactly.
Yeah, so there has been a lot of previous work in the lab, I think, with our some of our recordings that we do, instead of using a whole mount, we would mount the retina into an agarose solution, and then we can use the compressed dome to slice it in
Yeah,
different orientations.
wonderful.
Yeah, that can also help us decide if we want to record from ganglion cells versus maybe photoreceptors or bipolar cells. The orientation of the retina matters in that case.
Wonderful.
So with paired recordings.
Cool. Just a quick follow-up question to that, because we've actually had maybe half a dozen scientists reach out to us asking for tips and tricks on sectioning the eye, which is very tricky because they're so small. What are some tips and tricks you have for embedding in the sectioning process?
Yeah, so I think that question I would actually have to forward to some of the lab mates in my lab that work more closely with the retina, because a lot of my work does really center around doing the brain slices
The brain stem.
themselves.
Okay, wonderful. Thank you. So I wanted just to open it up and see if anybody else had additional questions. And always feel free to reach out to me, and I can put you in touch with Katherine and also the Singer Lab if you have questions later on that you think of. With that, I will wrap it up. And thank you so much, Katherine and Dr. Singer, for joining us and doing our Precisionary webinar. We greatly appreciate it. And we hope to see you, Katherine, in a few years at the next stage of your career.
Yeah, thank you so much.
Thanks.